special pleasure for me particularly to present our today's Heller lecturer, Professor Jean-Pierre Changeux from Paris. And I know that we keep telling our students that the next generation neuroscientists should combine thought with experiments, planning with molecular analysis, but there is already an example like that. Jean-Pierre Changeux, some of us still remember his work with Jacob and Monod in the field of proteins with the concept of allosteric interactions. And then he became a leading figure in the field of cholinergic transmission, both at the physiology and the molecular and the structural level. But at the same time, he's also a major figure in conceptual thinking about brain functioning, which will be our topic today, and a popular author of books that are vastly appreciated by people worldwide. Plus, he is a connoisseur of art. And I think this should be our role model in what a scientist should be. Not everyone can. And we are very, very happy that you accepted our invitation to give us a Heller lecture, and I'd like to present you with this.
Thank you very much. Thank you. Thank you very much, Hermana, for this invitation and for the privilege I have to give this Heller lecture. I hope I am not going to disappoint you, because I'm going to speak about a subject which is related to consciousness. And this is based on a series of papers, and you may find the presentation of all what I am going to talk about in... you think I should... Okay, so this is for your conscious perception of what I am going to say. And this is, in fact, a difficult theme, and I realize that the use of the word consciousness is forbidden in some disciplines, accepted in others, and at least by the referees of the review we wrote together, Stanislas Dehaene and myself, they asked us to eliminate the word consciousness from all the review. But they accepted one word, which is conscious processing. So I will try to discuss with you some issues about conscious processing, and more specifically within the framework of what we called the Global Neuronal Workspace hypothesis. So I will give a few definitions about conscious access, some modeling about the neuronal workspace hypothesis, then how to test this model with humans and with mice, if I have time. And what I'm going to talk about is summarized in this review in Neuron of 2011. And also I wrote several years ago some paper which was a little bit anticipatory, but not fully at the what we know today, but it's in 2006 about mice and how to approach this issue with mice, which will be the last part of my presentation. So from the beginning, I want to give the names of those who have been the main actors in this work. First is Stanislas Dehaene, where we met in 1986. He was 19, and our first paper was published at that time, modeling on bird songs. Then we continued to build models until in 1989, as you shall see, 98, we published this Global Neuronal Workspace hypothesis, and we are still working together, even though he is now bigger than I am, if I may say. He's a professor at the Collège de France. He's well known throughout the world, but we still keep going. This is, if I may say, the tradition of the Pasteur Institute to have collaboration between the advisor and the student, sometimes for a lifetime. So this is a very important point I want to make first. Then you shall see, if I have time, that we have been working on the case of the mouse, even though, of course, the issue of consciousness in mouse is a controversial one, and yet I want to show pictures of those who contributed to this work. Now, I would like to start by some philosophical point of view, which is tinkering models of the brain, which we have been doing. And our aim is to represent a behavior or a mental process, I emphasize mental, on the basis of a minimal yet realistic neural architecture and activity patterns, if possible in mathematical terms. I think this is not new for most of you, but I want to emphasize that. And the second aspect, which I think is an interesting, if not original, feature of this attempt, is to build a formal organism which is able to pass a task, a well-defined task. And with Stanislas, we have always been modeling with either the human or the mouse brain to fit with given behavior and to have predictions about the modeling we were doing. And we wanted more, which is to establish a causal relationship between a specific behavior or mental subjective process and objective neural measurements. And all this, and in particular the issue of this formal organism, should be challenged experimentally. And I want to insist on that from the molecular to the cognitive level. And again, I re-emphasize it and insist on that for a well-defined experimental task. So this is putting us in a very humble situation, if I may say, which was already recognized by the Greek philosopher Democritus, that the best model, who was a very strong materialist early in that times already, that the best model will never give a complete and exhaustive description of reality, which I think is something to meditate, simply because if we made a good model of fire, the computer is not going to burn. And perhaps we may hope that in the future, if we build a formal and hardware automat, it will able to behave in somewhat conscious manner. This is something which is still open to discussion. And just to say that we have never aimed to give a final answer about access to consciousness or conscious access, if you ban the word consciousness. Now, the second point I want to make from a general philosophy of all our work is that if you work on humans, you have to deal with several levels of organization. And these level of organization originate from evolution. And this is somewhat different than the levels that have been discussed in the computational community. This is more a biological view, which emphasize level of organization. And you may have other views about that, but let's say that there is grossly the evolution of species at the human genome level from the Australopithecus up to Homo sapiens, which is taking place in millions of years. Homo sapiens came out 100,000 years ago, which is a very short time, while Homo habilis or Homo ergaster lived about 2 million years ago. So it's a very fast evolution at the gene level. Now there is also another nested evolution, which is taking place during ontogenesis of any individual subject, and with stabilization and loss of nerve cells, but also, most of all, from the selection of synapses in the developing brain. This is not the issue of my presentation. I will give a more detailed description of this modeling in the seminar, which is going to take place in Technion or Symposium. And according to this view, the growth of synapses in the brain is taking place in a sequential manner. At some stage, which are described here, there is some kind of exuberance of connection, because all these axons are recognizing the same category of cells, and the state of activity of the network is giving a selection of some connection and elimination of others. So this is what we call with Antoine Danchin the selective stabilization hypothesis, which is of course essential for the imprinting of cultural circuits in the brain. And of course you know the cultural circuits for reading and writing, but there are many other ones which are being let down progressively from birth, even sometime before birth until the adult stage. And you have to recognize that this is lasting for 15 years and continues afterward, of course. But the Homo sapiens was selected 100,000 years ago through Darwinian evolution to spend about half of the actual lifespan of Homo sapiens in these early times. I think the human individuals were living about 30 years, and this is unique in the world of living organism to spend so long time to make the brain and to use it for such a short period of time. Well, yet that's the way things happened. And you can see here that as a consequence, we may have this kind of scheme for establishing some hierarchy from the molecular to the cognitive level, and that, of course, all of our higher production in our brain are rooted in this molecular or even atomic level, and building circuits from neurons and neurons from higher organization that I am going to discuss in my presentation. Now, there are additional, if I may say, Darwinian epigenetic evolution. One is the dynamic of thought. You are already in it, since you are already criticizing me about what I am going to say, or what I am saying, whatever. So there is some kind of selection of representation taking place in your brain. And of course, there has been the social and cultural evolution with testimonies which have been laid down by human beings throughout this past, which is even shorter than the gene evolution, of course, and where there have been extracerebral memories laid down, like books, like sculptures or paintings, since Lascaux and others. And this actually is taking place through the 100th millisecond time scale of psychological processing, and up to, of course, thousands of years. And I think we have to think about our brain, even our brain as scientists, as being, in fact, the synthesis of all these multiple evolutionary processes which are put down together at our birth, and then subsequently through these epigenesis, which is incorporating into our brain the social and cultural evolutions. Now, all this is consistent with a view of the brain, which is not the classical, I would say, input-output kind of model of the brain, which is classical in informatics, because since Shannon is always used as the brain as a black box with which is producing output from inputs. And I may say that from all this and some other arguments I may develop, our brain is exactly functioning the opposite way, from the inside to the outside. We are constantly projecting are representations that you are elaborating in our brain to the outside world and testing them. I think I see your eyes moving when I speak, and you are doing that during this period of time. You know, it's not a passive, the brain is absolutely not a passive sponge which is plastic and incorporate anything from the outside world being projecting to it. It's exactly the opposite. We are, in fact, constantly interacting with the outside world and integrating responses from the outside world into traces in our brain. So this is, after a few general statements, a slide that you may or may not like, and some of you may see the face of a man. Who sees the face of a man? Well, a significant fraction. I hope the other one is seeing the body of a woman. Well, a few one. Oh, very good. And some of you may even react and say, I see both. And indeed, once you have been accustomed to this Salvador Dali painting, you may, in fact, see that each two possibilities pop out. And the only thing I want to stress here is that there is some kind of processing of this image, which is a well-known case of ambiguous image, where only one representation at a time is being passing through our eyes and taking place in our brain. So it means that there is an all-or-none aspect in conscious access. And this is something I may wish to further discuss later on. And this painting just illustrates this point. And we have to find an explanation for that. So this kind of pictures just illustrate what we would like to approach. I don't think I can say more than that, which is to try to define conscious access from what you just saw in the previous slide. It's a subjective space. We have to start with that. There is in our brain, the subjective space, some kind of internal multimodal milieu, which is the case here. Since you are listening to me, you are trying to integrate all together the image you see but on the screen and what I am saying and all what you have learned before. So there is some kind of global workspace, which was defined by bars, where actions are received by simulations, replaced by simulation, plans, goals, and course of actions. And, of course, with the aspect of perception that I mentioned before. And in these global workspace, these simulation or plans are evaluated. So there is an evaluation process. I'm not going to discuss about this, but this is for future modeling to do in a global manner with reference to the outside world, the self, personal memories, internalized rules, and social conventions. This is some kind of a beginning of a definition of this kind of internal milieu that we have necessary in our brain in nearly all situations where we are awake and conscious. It's a poor definition, I agree with the critics, but at least it's a beginning. And what I want to further say is that we are concerned by what I call conscious access or conscious processing, which is taking place within this subjective space. And I would like to use the word conscious content to just specify what you saw in the Salvador Dali's line, either a woman or a man. So it's a conscious content. And you can say to your colleagues, well, or raise your hand and say, I saw a man or a woman. It means that there is a reportability of what has access to consciousness. And this is really one of the main features of conscious access. And I don't think you may omit this aspect in the definition of consciousness. I don't want to say it's unique to it, because you may have some case of reportability taking place under non-conscious condition, let's say, when you awake from dreams or something like this. But to my opinion, it's a very critical aspect of consciousness, which is to report verbally or by an intended gesture what you did of this current conscious content. So this means also, this is an important aspect that I am not going to deal with very extensively, is that there are other processings which co-occur with conscious access and which are non-conscious. And what I want to raise here is this challenge. Can we discuss a relationship between conscious and non-conscious processing? Can we establish a distinction with them experimentally and try to build some neural models of that? So this is something which is raised as a simple thing. In fact, it's not that simple, as you shall see. But we may tackle this issue as scientists and try to do the best we can. So we have also to make two basic distinctions in addition, not only to distinguish the content of consciousness, but also the state of awareness. And maybe some of you are already sleeping in the back. I don't see them. But they are in the state of consciousness, which is different from those who are awake and have their eyes moving. Well, I think I don't see anybody sleeping yet, but maybe it will come. But so this kind of state is, of course, taking place during wakefulness. Sleep, it's a different state of consciousness. Dream, it's another state of consciousness. Of course, general anesthesia. I think many of you may have been operated and have been undergoing general anesthesia. And there, the use of the word consciousness is extremely accepted because all anesthetists say, oh, he has lost consciousness. Oh, he has recovered consciousness. So this is something that in the field of general anesthesia, it's a commonplace. Coma is, of course, another state of consciousness, epileptic crisis, and for some people, orgasm. I hope many of you. But the second aspect, which I mentioned before, is the content of consciousness, which, as I said, is the thing that we are going to be interested with. So, first, to try to show one slide about what I am not going to talk about, and which nevertheless is a very active field of research, and which is the control of these states of wakefulness, sleep, dream. So there have been a huge amount of studies in the past 50 years about this, and I want just to mention one point from Israeli scientist, Devor, who did a beautiful experiment by injecting pentobarbital, which is a GABA agonist, so it's stimulating inhibition, in a mesopontine tegmental nucleus, which is present approximately here. And the issue is to check whether the general anesthetics, I will come back to that, whether or the control of sleep and wakefulness is determined by a few population of neurons or a very large one. And this is a very important aspect for general anesthesia. And what he was able to demonstrate is that when he is inhibiting some of these neurons, there is, in fact, loss of consciousness, which is taking place in the rat as a consequence of this manipulation. So this is a very important aspect of the physiology and the molecular biology of the control of states of consciousness, and there have been a lot of studies, as I mentioned. Here are a few of the many workers who have been working on this. And these goes also with some observation from neurology, where a lesion on the brainstem, in the tegmentum in particular, which is a cholinergic system, then there is a loss of consciousness with coma. So these neural mechanisms for control of states of consciousness are, of course, very important and are some of the prerequisites of what I am going to talk about. And this is, of course, something that all of you working on brain modeling have to take into consideration. And imagine that you are not going to modelize brain during sleep. Well, you may, but usually when one is looking at cognitive function, one has to modelize the wakeful brain, and that's what I am going to talk about. So, what is our basic hypothesis? And I wish to say it's a very simple-minded one, and I'm pretty sure that some of you are going to be very disappointed because it's too simple. But at least it's something that we can use for additional work and demonstrate that it is incorrect or insufficient. The main hypothesis that I'm going to put forward is simply to account for this workspace, this milieu, where we have, I would say, a synthesis of sensory input to have plans and memories accessible altogether. This is part of the definition I gave for conscious access. So how to link in the brain all these different components which are in fact operating separately in the brain? Well, you could use some kind of waves or some kind of spirit or mind or whatever. And the things that us as neuroscientists have to look forward is whether there are some neuronal circuits for that. And the simple postulate that we propose is that within this global neuronal workspace, there are some neuronal connectivity which is accounting for the global character of the network. So just to give a few reference to the past about Descartes, I think any Frenchman should mention Descartes, that there is a limited capacity system in the brain, which is, I think, a very important aspect. There is, we can think about only one thing at the same time. This global workspace was, as I said, proposed by Bars, but the neural basis of it was not made explicit. And there was also this Norman Childish kind of model where there is some kind of supervisory attentive system and contention scheduling process, so two levels. And also the issue that maybe reafference or re-entry should be important. I'm going to discuss again these aspects and from Edelman, Lemmer, and Rolf Seemann and others. And our hypothesis is that to establish a global workspace, we have to rely on a special feature which is present on pyramidal cells in the cortex, which is the existence of pyramidal cell with long axons. And this was recognized by Santiago Ramon y Cajal very early on. And not only pyramidal cells which interconnect the front with the back and conversely, but also from one hemisphere to the other and have some kind of heavy interconnectivity within an hemisphere and between hemispheres through the corpus callosum. The other evidence for that is that from neurology, from Desjardins, who was a very distinguished French neurologist at the turn of the century, and who identified these fascicles of white matter in the brain, in the cerebral cortex. And he distinguished several of these fascicles of white matter, and it's very interesting that diffusion NMR or tractography done by Pugliese and colleagues, which is shown here, has identified exactly the same long-range fascicles. And you may already notice that most of them, not all of them, but most of them originate from the prefrontal cortex. And this is, of course, an important observation that we have to deal with, and which has been missed by most of those who have tried to look at hubs within the cerebral cortex, because they are only looking at nearest neighbors kind of connectivity. And the other aspect which is important, and which goes back to von Economo, who was a follower of Brodmann, is that if you look at slices of cerebral cortex, you find that these pyramidal cells are from layer 2 and 3 from the cerebral cortex, and that they are most abundant in type 2 cortex. And this type 2 cortex is present here, as you can see, in the dorsolateral prefrontal areas, also in the parieto-temporal cortex, and in the cingulate cortex. Moreover, if you look at the work of Patricia Goldman-Rakish with the monkey, you can see that she found a network of long-range connection, which is interconnecting the prefrontal areas with the parieto-temporal cingulate cortex as well. So clearly, there is some kind of internal topology within the brain, which is I would say pre-organized, and this is a fact. Now, the hypothesis is that we relate this long-range connectivity to the global neuronal workspace. So this is the hypothesis, and of course, this kind of long-range connectivity will favor broadcasting of signals to multiple brain areas, and conversely, just yielding subjective experience. So this is the hypothesis that we want to test. If I may say, it's a very simple-minded one, and it is something that is accessible to experimentation. I hope I will convince you of that. Now, if you look at the evolution of white matter from rat to humans, you will always think, oh, he has a lot of gray matter, which means that he is very intelligent. Well, I would prefer to say he has a lot of white matter, which means that he has a lot of interconnectivity in the brain. And if you look at the evolution of white matter, you can see that there is a dramatic increase of white matter, which is taking place through evolution in the brain. And here you can see that there is actually a shoot of development of white matter compared to gray matter. And this is from the work from the famous German neuroanatomist Carl Zieles. And this clearly shows that if something is evolving in our brain, in addition to the increase of the surface of the cerebral cortex, is the considerable development of long-range interconnectivity in our brains. So this is in fact a fact, and there is, of course, something which is correlated to that, which is the expansion of the prefrontal cortex, which is taking place throughout evolution. And as I said, layer 2 and 3 neurons are more abundant in the prefrontal cortex, so that when you increase the prefrontal cortex, you increase the long-range connectivity, aware that it's not a unique source of the long-range connectivity, of course. Now, this is some work done by Elston on these, precisely these neurons from layer 2 and 3, which are long-range axons. And he has been studying quantitatively the basal dendrites of these pyramidal cells. And you can see that indeed the complexity increases up to the prefrontal cortex. So there is also a species dependence there, and that when you look at the complexity of these dendritic arborization, it's of course increasing very fast in the prefrontal cortex, and here for human, macaque, and marmoset. So it's clearly something which is evolving at a very fast rate in the course of mammalian evolution. And you can relate this expansion to the relative expansion of the prefrontal cortex compared to the rest of the cortex, which is, of course, the highest in the presently living human beings. So this is just to illustrate that one can correlate this anatomical work with the postulate we have made about the neural basis of the Global Neuronal Workspace. Now, this is another view of the hypothesis we proposed in 1998. And the distinction to make some network simulations, the distinction between, I would say, specialized processes following bars, which are here for instance, visual or auditory perception, motor actions, another long-term memory system like the self, which is, of course, something very important, and the language areas, somewhere there. And then, so the idea is that these are specialized processes which are modular, encapsulated, and automatic. And then the global neuronal workspace with these long-range axons would, in fact, exist on the top of this set of processes and behave as a top-down kind of system regulation, and where this issue of reportability would occur, thanks to the fact that if there is broadcasting all over the brain, then of course we may report using language from our conscious experience, because there is a common space for it. And you can see here that we can view the representations which are taking place within the global workspace, the conscious representations, if I may say, which are made up of these neurons from the I just mentioned, plus those which are being collected from the processes, and which give, I would say, the specificity of the representation. It can be a visual, auditory, and anything like that. So this is the general hypothesis that we put forward for modeling. Now, what we have developed for neurocomputational studies are extremely simple, of course, and I'm going to present them later on. And I want to say that this is, again, very simple-minded. And in the next slide, I am going to show some experimental data which has been done by Stanislas Dehaene in the CEA in Paris at Neurospin, and which have been devoted mostly to try to test this hypothesis. Now, to do some kind of to have an experimental procedure to distinguish between conscious and unconscious processing, which is in fact what we want, one has to find some kind of task, as I said, and what we are going to do is to try to represent by a neurocomputational model how to account for this task. And here you have a very simple task which is called a masking task. It's an old kind of task which was already developed in the 80s, where you present to a subject a sequence of slides here. Here a mask, here an empty slide, then the word lion, then an empty slide, then a mask. And when you can look at the dynamics of it, it is in the 70 millisecond time scale. And when you ask the subject, have you seen something? Yeah, he said, yes, I have seen. And he answers, or she answers all the time, lion in French. So it's a typical conscious processing. Now, if you do the same experiment with the same slides, but you change the order of the slides, and here you put a mask instead of empty slide, put the word note, and so on. And then you ask the subject, have you seen something? He said, no, I have not seen something. But if you ask the subject to select the word note in a list of words, then there is a preferential selection for note. So it means that the word note has been processed by the brain, but in a non-conscious manner. So we have here a situation where we have a task where you can distinguish between conscious versus non-conscious processing. And then this is a subjective experiment for the subject who is undergoing this task. And now we want to have some objective parameters of what we can see in the brain. And this slide shows, it was done early by Stanislas and his team in 2001, that if you compare the brain of a given subject under these two situations I just illustrated before, you can see that there is a dramatic difference between the two. And in the case of non-conscious processing, the visual areas are being activated, the primary and a little bit the secondary. And here you can see the left fusiform gyrus, which is involved in the recognition of words. But most of all, you can see that in the case of conscious processing, you have some activation where exactly at the location where you anticipate to have the global neuronal workspace. So it's a pure correlation, I want to say. It's not a causal kind of situation, but at least there is some relationship between this long-range network that I illustrated before and the conscious access. Now, this is not a unique experiment. It has been repeated by other groups and in parallel with Stanislas, even sometime before, sometime after, whatever. I'm not going to give priority to anybody there. For us, which is important is that you can have the similar situation for sound, and you see this is, and for motion, intentional motion, in a go, no-go kind of task. So you can see that this is not unique to the visual access, but also to other modalities. So this task is can be used as a model for conscious access. Now we have used, I should say, Stanislas has been using a more sophisticated paradigm where you present this succession of slides here, and where you have a T1, which is KOOX, and a T2, which is a second stimulus, which is 5 in French. And then you ask the subject whether he has seen or she has seen or not the word 5. And the interesting thing is that this is some kind of task where the report is not a gradual one, but it's an all or none one. So either the subjects are able to report 5 or not, depending on the time lag between T1 and T2. So there is a simplified and more precise paradigm than the one I illustrated before, where you have a first stimulus, then a second stimulus, and a time lag between the two stimuli, and you ask the report. And in some case, when this time lag is too short, then the subject is not being able to report T2. So what about the EEG kind of recordings you can get when there is visible words and masked words? You can see that there is a very striking difference which is taking place around 2 and 44 milliseconds. That here you have a very broad invasion of the prefrontal networks, which is not taking place in the case of the masked words, while you have, in fact, the early stimulation of the visual areas, which is taking place non-consciously. And when you continue the experiment, you see that there is a persistent activation, which is taking place beyond 400 milliseconds, and that the main critical event being around 200 and 300 milliseconds, when there is this what we call ignition of the cerebral cortex during conscious perception. So this is a very striking kind of objective, if I may say, attempt to get some physical parameters about conscious versus non-conscious processing. Now, this has been further studied by Stanislav's group, as well as other groups. This is just to illustrate the data that were published by Sergent and colleagues. And you can see here that during conscious processing, there is a sharp wave which is taking place here beyond 200 milliseconds. This is just different EEG recording as a function of time, just what I showed before. And so there is a clear, sharp, I would even say, all or none kind of phenomenon which is taking place around 200 milliseconds, and which is accompanied by some kind of waves, which called P300 or P3B waves, and this is taking place in the prefrontal cortex and not in the middle temporal cortex here. So you can see that there is some kind of selectivity for the prefrontal global network. Further studies have been done by Nakash with patients, where you can have intracranial recordings being done in epileptic patients, and you can see that there is clearly differences in the visible and invisible processing of written words, and you can also see that there is in the gamma band power a very significant change between the two, as well in the beta phase long-range synchrony, which is much stronger in the case of the visible versus not visible situation. So there are further additional processings which are being studied, and parameters for that, one being not only the EEG response, but the synchrony, which can be recorded from EEG and from MEG experiments, which have also been done. I want to say that these are not the only one experiments which have been done in this field, and Rafi Malak and Isaac Fried, who is working, I think, here in Hadassah University and Tel Aviv. I'm sorry, I made a big mistake. In Tel Aviv University, and that you can have the same thing in humans by single cell recordings, and having the clear distinction between the twin towers in a subject when he recognizes or she recognizes the picture and not. So this clearly shows that there is also a threshold, and if you remember well, from the painting I showed by Salvador Dali, I emphasized this aspect of an all or none switch. So this is something which is indeed happening following objective recordings of conscious access. Now, this is the kind of simple-minded scheme that we can propose for access to the global neuronal workspace when it is subliminal or non-conscious processing. You activate only the visual primary or secondary areas, and then when you have this conscious access, then you mobilize the prefrontal cortex and the global neuronal workspace circuits. Now, we have tried in a paper written in 2003, it's a little bit old, 2005, it should be reactualized now. We try to modelize this conscious access on the basis of spiking neurons and classical cortical columns. And here we have, let's say, the T1, you remember, and the T2. And we can follow the processing in a feed-forward manner. And again, it's a very simple-minded kind of model, but at least it's a model. And what's happening here is that either the system goes up to give global ignition, or not. And the very interesting thing is that there is, of course, lateral inhibition, which is taking place here, and that to have, in fact, the all or none switch I just mentioned for access to consciousness, you need some kind of feedback connectivity, which has not exactly the same pharmacology as the feed-forward. To simplify, we said it's AMPA receptor here, an MDA receptor, it's slower. And you need altogether the feed-forward and feedback connectivity to get this ignition of the global neuronal workspace, and then have access to consciousness. Again, it's a very, very, very simple kind of scheme. It could be made much more complex, of course, much more elaborated, but at least it gives an interesting clue, because it put an emphasis on the retrograde feedback connectivity or reafference. And as you shall see, this is what we think is the privileged target of general anesthesia. Now, these are just the simulation which were published about the ignition within the global neuronal workspace, and the fact that if you have a blinked trial where the lag is shorter than in the case here, you have, as I say, masking which is taking place, and the ignition is not taking place anymore. So this is, if I may say, not really something extraordinary. It's a very simple kind of scheme which can be further implemented in a more concrete or more explicit terms, and trying to do further work on this conscious access in the brain. And just to illustrate this different step of conscious access, to have the early visual processing in V1, here around 170 to the extrastriate and fusiform cortex, then the first contact with the workspace neurons in the 250 milliseconds, and then the global ignition and its long-lasting perpetuation, because that's a very important aspect of it, is that these representations in the brain are long-lasting and have this happening if, of course, the simulation is taking place under condition where the threshold for ignition has been reached. So this is just to summarize the present state of the situation, which at least gives a hint of how one can approach the issue of conscious access using objective parameters. I think this is for us a very important aspect of all this attempt, is to have ways of following conscious access in the brain and non-conscious access and processing. So to have scientific, objective measurements being used for a well-defined task. Of course, there are limitations to this approach, but at least it gives us a minimal, I would say, objective approach to the issue of access to consciousness or conscious access. So I am not going to repeat what I just said. There is this all-or-none amplification, the late P3B wave, the late amplification, and enhanced long-range phase synchronization, particularly in the beta range. So this is at least the first step. And now I am going to go into some clinical applications which might be of use for all of us trying to do models, and maybe one can try to find some task which may use patients to test whether they have some deficits or not in conscious access. And of course, one of the first one is general anesthesia, I just mentioned already. And if you look at fMRI, there is a decrease in blue of the activity in the prefrontal cortex and here in the parieto-temporal, but it's not very clear. Yet, if you do a more rigorous experiment by electrical recordings that you can do by EEG for surface activity and using deep electrodes here, which are implanted in these epileptic patients, and just submit these patients to general anesthesia, I think under ethical condition, which has been approved by an ethical committee. Of course, this is a very important aspect. You can see that there is an all or none switch, which is taking place in the cortex, compared to a very slow evolution of the subcortical activity, which is recorded by these intracerebral electrodes. So this is a very nice experiment from Vély and al. From Marseille in the south of France, which clearly show that there is a contribution of the cerebral cortex in this state of general anesthesia. And what are the sites of general anesthetics? I'm not going to give you the list of all the sites. I just want to say that we have been recently working with bacterial receptor. And you may say, what's the relationship between bacteria and general anesthesia? Well, the relationship is that both bacteria and the human brain are sensitive to general anesthetics. And the thing is that from the work we did in the lab, mainly by Pierre-Jean Changeux here and Marc Delarue, one has been, and from the work which was done previously on pure genomics studies on bacteria, indeed bacteria are able to produce receptors which are highly similar to the nicotinic receptor. If you know this 3D organization of the nicotinic receptor, you will immediately recognize the similarity, and the homology is at the level of the tertiary and quaternary structure, much more than the level of the primary structure. And the very interesting aspect of it is that if you look at the effect of general anesthetics and where the general anesthetics do bind to this receptor, you find that it's binding to a site which is located in the transmembrane domain, which is also a site for what is called allosteric modulators, which have been identified with the nicotinic receptor, and that this site is nearby a lipid molecule. And this is for us a reason why we think that these receptors are sensitive to general anesthetics, and why general anesthetics are so efficient in the brain, is that they are acting not only on GABA receptors, as you know, but also on nicotinic receptor, either as positive allosteric modulators or negative allosteric modulators in the case of the nicotinic receptor. And very interestingly, this is from the work of Jonathan Cohen and colleagues, you find that the site for general anesthetics, which is here etomidate, on the GABA receptor, is located very nearby the one for general anesthetics in the bacterial receptor. So we think we have a hint to link the molecular biology to this higher cognitive function, which is consciousness, because there has to be some kind of relationship between these molecular targets and the global neuronal workspace. So this has been investigated by several groups, in particular by Mashour, and using some kind of manipulation of the EEG data using symbolic transfer entropy as a measurement. And he has been able to distinguish between what I call, you remember, the feedback and the feed-forward processing in the cerebral cortex. Remember what I showed and the model I presented. And the very interesting thing is that, first of all, the feedback connectivity or feedback activity is more important than the feed-forward in the resting brain, which is okay, since in the resting brain it means that there is a lot of spontaneous activity which is taking place, which is something which has been recognized for years. And that when you give the general anesthetics, you have a very differential loss of the feedback processing compared to the feed-forward one until the two become equally diminished. And when you have recovery, you can see that the recovery of a conscious access for the anesthetist is the recovery of consciousness is taking place mostly, according to Masho, when the feedback connectivity is functionally restored. So these tend to show that there is some kind of preferential aspect of general anesthetic effect of general anesthetics on these feedback connectivity. And to go along these lines, Lame, who was also being important in studying this feedback, top-down feedback connectivity, showed that if you give GABA agonist to the subject, it's again something which is a little bit at the fringe of ethical experimentation with humans, but I think many of you may take, in fact, allosteric modulators of the GABA receptor with benzodiazepine and so on and so forth. So this is acceptable, and you can see that it's reducing visual awareness by inhibiting late feedback activity. So there would be, in fact, targets for general anesthetics and modulator of the GABA receptor, which affect selectively the top-down long-range connection at the level of cortical GABA receptor. We know that there are a lot of inhibitory neurons in the cerebral cortex next to the pyramidal cells. And of course, if there are inhibitory neurons, there are also GABA receptors. And therefore, this is a possibility to further investigate. So just to say that this is an interesting kind of system which can be studied with animal species in addition to humans. So now, what about schizophrenia? This is an experiment done initially by Rodriguez and Varela in France in the late 90s, before his unfortunate death. And the experimental paradigm is the following, where you show a face which is called a Mounet picture. Has nothing to do with the moon, but with Mr. Mounet, who invented the experimental paradigm, where you put upside down a face, and for reasons we may discuss later on, these human beings recognize face on the right position and not on the upside down one. And this is something which has to do with the perception of gravity and the fact that we are looking at faces on always. Even for the newborn child, there is something there. So I am not going to discuss that. But the interesting thing is that it could be used as a very useful task to follow the phase synchrony of EEGs. And you can see here that here in the beta band, there is a beautiful activity there in the normal subject for the faces being recognized and which disappear in the case of non-recognition. This goes back to the picture I showed of Salvador Dali, if you remember before. So the same kind of measurement could be done. And here, if you look at schizophrenics, there is a very striking loss of organization of the response in the beta band range. So there is clearly a deficit in the schizophrenics in conscious processing. So our view is that perhaps there is some altered global neuronal workspace processing which is taking place in schizophrenics. What about the development? And again, this is now from still Rodriguez, but with in the lab of Wolf Singer. And what he found is that in early adolescence there are some traces, very significant traces of beta synchrony. Same thing in the adults. But very interestingly, in the late adolescent, from 15 to 17, there is a dramatic loss of conscious processing. Well, this may be some kind of neurophysiological explanation of some problems in the behavior of adolescents that we have to deal with. And instead of putting them in prisons or letting them use drugs to stimulate their own Global Neuronal Workspace, we perhaps should take care of them more carefully. And this is something which is interesting to note for the social implication of these studies on the brain. And this is also an interesting aspect of to explore in the case of autistic children. And then the diffusion tensor imaging tractography method that I introduced earlier on revealed that there are significant differences in the long-range connectivity of autistic children. I don't want to say that the whole explanation of the autistic brain, but it's taking place, and instead of having a reduced connectivity that you may find here, you have an enhanced connectivity, which means that there are too many connections. And what I introduced as a selective stabilization hypothesis taking place during development is not taking place correctly there. And there is more connection than necessary, and this is, of course, perturbing the processing of conscious representation by the autistic children. So this is still something to further explore. So I give you some hints where I think clinical application could be used to develop programs on conscious access. Now there is, of course, the frontal lobe dementia. I hope I am not going to finish on these states, but it may happen in a large number of elderly people. And you can see that it's characterized, it has a genetic basis. It's characterized by a dramatic loss of layer 2 and 3 neurons, which are those which are involved, according to our views, in the global neuronal workspace. So this is something that we may have to take into consideration. Now, I think it's already 6:10. I may perhaps stop there. And I just go to the conclusion. So just to say that experiments can be done in mice with the nicotinic receptor, and just to test these views. And what I'm going to say that here is that, of course, one should not consider that this approach that we have used is going to give a full explanation of conscious processing, because there are, of course, levels of organization and levels of evolution of consciousness, and that, let's say, a rat or a cat has not the same development of the access to consciousness as an adult human being, and in particular there is self-consciousness, which is, of course, missing at this early stage. So it means that there is more work to do there, and we can objectively continue along the line I showed before. And the conclusion is that we have still many unknown about access to consciousness. There are other aspects which is called metacognition, which is something that we are able to judge about whether we are conscious or not. So it means that we are conscious of being conscious, and so on and so forth. So it's just a start, no more than that. And I may say that I will finish by showing you, just to please Hermona, a manuscript which is actually in sale at Christie's, if I remember, which is for Mazor from Firenze, 1490. I just show it to see how these beautiful pictures have been actually painted according to the Jewish tradition, even with human pictures, because we had a debate together about that. Thank you very much. Yes.
What do you think is the main role of the feedback instrument? Is it more to enhance the sensory in order for the concept to be conscious?
Yes.
Or is it
according to our model, we, and this is also a Lammer hypothesis, is that it is necessary for conscious processing. It's part of the neural system or neural network which is giving this all or none access to consciousness. And I emphasize that because I think this is one of the key characteristics. Again, it's a very simple-minded kind of view. But many of the models which have been done in neural networks are done without this aspect. And one may easily have further modeling of conscious access using this aspect of it. And interestingly, it looks to be the target, or differential target of general anesthetics. And so my opinion is that there is a causal relationship between the presence and mobilization of this reafference. You know, it's an old concept, the issue of reafference, and which is, of course, very general in brain processing, and in particular for conscious access.
Jean-Pierre, what is the objective basis for the statement you made that in autistic patients or patients with Asperger syndrome there is a lack of selective stabilization? Why not perhaps aberrant type of connections and stuff like that, which do not evolve from a lack of...
The reason is that, and I have a very good answer to that, is that recently some of the pathways which are involved in synaptic eliminations have been investigated, and they have a lot of sequences from the receptor downstream. And one can find that some of the genes which are mutated in there are many genes, but some of them are affecting selectively this selective elimination process. We had a seminar at the Pasteur Institute 2 weeks ago about that. So I think there are additional objective data about this. Now, I think this selective stabilization is a very widespread kind of phenomenon which is taking place. And the fact is that the approach of the afferent axon to their target is somewhat imprecise at the beginning, and then there is ultimately tuning up of the connectivity which is taking place. And this is very general for the development of the brain in general. And this kind of phenomenon, which is we presented it in 1976, too early in the sense that it was very popular at that time. We have about 800 quotes or something like this. And now people are not really assimilating that, which is a pity, because I think it's absolutely crucial for brain development and for, in particular, the non-linear evolution of the genome compared to the brain complexity. You know, the evolution which went from chimps or common ancestors up to humans does not involve many change at the gene level, only a small number of them. And the rest is being done by processes which are existing in the brain. And in particular, this phenomenon, which, to my opinion, is at the beginning of the non-genetic kind of organization. It's within a genetic envelope, but it's epigenetic. I use the word in this sense. Now it has been used by molecular biologists in a different meaning, but I use it initially as such, but they simply ignore what the work we had been doing before, and now they use it for modification of chromatins and so on and so forth. But I think this kind of processing is absolutely fundamental, to my opinion. So it's not against some kind of universal feature about the organization of the human brain, which is genetically encoded, but it gives. I had a discussion this morning, which was a little bit agitated, if I may say, between universal ethics and communitarian ethics. You know, and I was, of course, defending a universalist position, and the argument I used was, of course, the human genome and the main feature of our brain, which are common to all different ethnical groups in the Earth. So we may discuss that later on. But just to emphasize the importance of epigenesis, and also the fact that give an interpretation of the diversity of cultures, because it's based on it.
What do you think might have been the effect of, you mentioned cultural evolution, but especially language?
Yes.
Language appears to be a very powerful factor in consciousness.
Of course, I think this was emphasized also by Jerry Edelman, just to be fair, and we have not dealt with this at this stage of modeling, but I agree that it is a very important feature that we have to take into consideration in the future for high order consciousness. But we have to distinguish in the framework of evolution between oral language and written language. And
oral language, but also only 10,000 years old.
No, but I think the difference is that oral language is to some extent predetermined in the brain. There has been some kind of course, the Broca and Wernicke areas are to some extent genetically encoded as such. The ability to learn language, if I may be more specific. And that, on the other hand, the written language has been invented by humans in Iraq a long time ago, 8,000 years ago, I think. And this discovery, which was an invention of written language, the interesting thing is that thanks to the epigenesis, some aspects of the environment which were encoded in our brain has been exchanged with this kind of representation of the world, and that from Desjardins, who was really a genius at the turn of the century, it is well known that there are connections in the brain which are being mobilized for written language exclusively. And he showed that there are, for instance, alexias without agraphia, which means that some lesions in the cerebral cortex give a loss of the ability to read but not to write, so that people are writing without reading what they write. And so it means that there are cultural circuits which are due to the environment, which are built in our brain. And this is true for other things, of course, fortunately or unfortunately, in some cases, as you may imagine. I don't hear you. I'm sorry. You are talking about schizophrenic patients and autistic children. Well, the fact is, it's a fact that they are cut from the outside world. You know, this is something that is very characteristic of schizophrenia, is the hallucination. There are some endogenous voice, like Jeanne d'Arc in France, who had voices, and many other people. But but in the case of schizophrenia, is really pathological because they are really suffering enormously from this. We should not smile about it, but it's a very painful kind of feeling to have voices, inner voices, telling you to do that and that and that. And the fact is that they cannot correct the spontaneous endogenous hallucination through the interaction with the outside world to just adjust their internal thinking or whatever you call it with reality, which is outside. So there are clearly perceptual deficits which are taking place. Yes.
I think the question is was there psychological evidence about perception apart from the brain mapping?
Of course, there have been a lot of data on these perceptual aspects of schizophrenic patients. There are a lot of differences which have been noticed and which are extremely interesting to work with. You know, it's one of the most interesting application of brain science is to try to have ways of trying to approach the psychology and physiology of these patients. And if we can understand then, try to find remedies, which is something which all of us concerned by brain science should be aware of and try to find some ways of circumventing this kind of deficit, which is, to my opinion, extremely difficult to circumvent in adults. And I think the new treatment for schizophrenia is going to be on children with predisposition to schizophrenia, so that they don't express the phenotype.
I guess if I could just try rephrasing that question one more time and then ask my own. It might have been hard to hear from the back of the room, but I'm also curious. He was asking about if you showed a picture with the Mooney.
Yes.
The Mooney pictures where you
Yes.
showed a face right side up and upside down.
Yes.
And a difference in the objective brain
Yes.
response.
That's a very interesting task to modelize by those who are making computational modeling, I think.
What we're curious about is specifically about this image of a face. Did the schizophrenic patients...
No, I think, you know, as I said at the beginning of my presentation, all our task as neuroscientists is to find a way to find a characteristic paradigm, behavioral paradigm, so that we can progress in our understanding of brain functions. So we have to select a paradigm, and this is one very interesting paradigm. And it is known that the schizophrenics have a lot of perturbation about the perception of the outside world, but you have to specify it and to find a way, to find a task, as I said at the beginning, where you can assess something different, you know. And this is, I think, an interesting task which was developed by Varela, actually, very early on, and which is actually very... he made further studies about that. You can look at the early papers by Varela group and show the temporal processing of the oscillations, which are, among others, being correlated or not during the processing. And I think the early data that Varela are very consistent with some of the Stanislas findings. So there is no contradiction between any of these kind of experiments. They are on the opposite, a lot of coherence, if I may say.
May I ask about
yes, please.
Another part of the presentation? The part where you were talking about the effect of general anesthesia.
Yes. Yes.
You have located a certain part of the brain, but if you inject just a little bit of...
No, that was the first experiment I showed about Devor kind of finding. And just to be complete about that, and you are perfectly right that I was very short on this issue. But, you know, I have written a review on this. If you are interested, I can send it to you. The thing is that there are at least two main targets of general anesthetics. One is the ascending system from the midbrain, and the other is the cortex. Because you need to have, you know, the cerebral cortex in a state of awareness. You know, you have to be the subject to be awake if you want to do experiments about consciousness. So you need this kind. And then in the awake subject, you are looking at what's happening in the cortex during conscious versus non-conscious perception. And what I want to say is that most likely the general anesthetics are acting on both kind of aspects, but I discussed in my presentation only the conscious processing at the level of the cerebral cortex.
Thank you very much.